everybody. First of all, because I tend to talk too much, I want to say thank you to the people that have done the work. And this is a bit of a new talk to me because I'm presenting the iPS-derived neurons work and organoids work, which we started just a couple of years ago. So I'm a fly person. All the work that I'm going to point in the first few slides of the flies from people in the past, and some of the new work in human cells is with people in the present. And you can see the collaborators, which without whom I wouldn't be talking here, really. So I want to tell you, I've been interested for many, many years in cell and tissue organization. So I am a basic scientist. I don't study specifically diseases or therapies or diagnosis. I'm really trying to understand how cells and tissue organize, and also what may go wrong with that when a disease may develop. And I'm a developmental biologist, so I concentrated a lot on how cells and tissue organize during the very early stages of embryo. And I've been doing this with the fly for many years, and now trying to analyze some of this, both in developmental framework, but also in a disease framework. Before anything, I want to thank Rocco and Wider for many years of wonderful collaboration, amazing people to interact with. And I want to say that they are basically the power couple of Tunisian biomedical sciences, like Angelina and Brad, but even better than Angelina and Brad, because they are going to carry on doing this for many years. And I can see that why this guy done. So really wonderful. Now, what is the question in general? Oh, let's take this off. Okay. So basically, as you know, we all come from one cell that is fertilized, the egg, and that's it, right? So how do you go from that one egg that divides, self-organizing different cell types to very complex structures? Here's just the eye in the zebrafish, a beautiful movie that was given to me by Karen, where you can see how during organogenesis, in earlier stages of embryogenesis, this is all happening really early in human embryos, a lot of organization has to take place, a lot of cells and tissue. And one of the things with organoid field that it showed is that a lot of this information is intrinsic to that cell and that tissue. It's a self-organization. It's not always depending on signals coming from the outside. So you can take a stem cell, put them in a petri dish, add a few factors, which have helped, and they are able to self-organize into quite complex structures. In the fly, we've been studying this, as I'll show you, with a model with the ovariole of the fly. In humans, we are looking at both iPS-derived neurons for more adult behavior of the cell, and cerebral organoids for the early developmental stages of the brain. And the question, as I said, is really I'm trying to understand cellular self-organization, which is basically trying to understand how the interaction of the components of the cells are able to come up in a time and space regulated manner with complex structures that have to function properly. And as I said, I've been doing this for many years in the fly, and I'm now trying, and I'm new in this, so I'm actually learning doing the workshop itself. So I feel quite close to you in that sense, because I have experienced how challenging it can be, but also how beautiful it can be to work with 3D models. Now, why do I do the fly? I want to show you a couple of slides about this. You may know that Drosophila has been a model system for many, many years, and there are a lot of similarities, but also lots of differences with human. I'll show you a few examples. And what we want to use, what we've been doing is findings that things that we found in the fly, which is very inexpensive to work with, very fast, and you are working with a whole animal, a whole body, organogenesis within the whole body. Things that we find there, we are now trying to understand whether they work the same way in humans, basically. And I'm going to tell you two stories. One is regarding neurodegeneration, and the other is regarding epithelia columnarization, so the shape of epithelia cells. And I'll tell you those two stories. And again, I have a lot of data on flies about this and not so much in the human cells, as you will see. Now, why flies? I've told you it's easy, it's inexpensive, it's impacting, so you it's been used to understand fundamental principles of genetics and development, but it's also helping to understand some aspects of human biomedical questions and also human diseases. There is a huge number of genes that are conserved between us and flies, but also when they are not conserved, you can actually create a model, and it's been done for cancer, neurodegeneration, metabolic diseases, studies of behavior. The neuronal network behind that behavior is endless. Even if you don't have the conserved gene, you can make what is known as a humanized fly, where you express your human version. And you may develop a model which reflects maybe some aspects of the disease and not others, which is okay. So it's good to know the limitations as well. And that's what I want to say at the end, that there are differences. There are differences in the brain anatomy, cardiovascular system, respiratory system, but there are a lot of similarities. So know the limitation. And the work in fly has given rise to several Nobel Prizes, including the last one in 2017. Our main model system in the fly that we use, the main organ that we are studying is the ovary, but we have also done work in other systems, including neurons, as you will see. But in the ovary, we've been working, there is a lot of events happening during oogenesis, and they create an oocyte the same way that we do, very similar. You have the germline, you have the somatic cells surrounding. There is interaction between these two types of cells. There is a lot of polarization, all that, and a lot of migration. That has made the egg chamber a very good model for years and for several labs to understand morphogenesis and behavior of cell and tissue in a whole animal context, which is one of the powers of the fly. And things that happen at these 8 stages of oogenesis have been shown to be very important for embryogenesis after when the egg is fertilized. And I have done a lot of work, and I only want to tell you a couple of slides on this, on how the oocyte gets basically organized, how things move. So we study a lot motion of cytoplasmic components. How do they move? Where do they go? How do they go where they have to be? And which cytoskeleton these motions depend on? And there is several papers on that. And this is important not just to localize things somewhere in the cell. You know, we heard about exosomes, and the exosomes have to move out of the cell, and that is completely depending on cytoskeletons. So motion in cells is crucial for life. And that's what we are trying to understand. What type of motions are there in cells? What of these motions, which ones are important for the function, and which cytoskeletons are responsible for which motion in those cells? And there's a lot of work published. And I want to say that this is not only important for the function of a healthy cell, but it's also been linked very strongly to diseases, including neurodegeneration, as you will see a few examples. Regarding another thing that I was telling you, so this is the part of neurodegeneration that I want to tell you about in a minute. The other part, which is epithelia organization, we basically have 3 types of epithelia in our cell: columnar, cuboidal, or squamous. And it's not really understood how the epithelia cells adopt one shape or another. So we've been studying this for a while. And of course, that epithelia shape is important for the organogenesis, for formation of the organ, as well as for the function of a mature epithelia later on. And we've been studying this in the fly for a while. And again, now we're trying to analyze what may be true in the human models that we are looking at. So that's just bringing up the summary of what we've been doing in the fly. Regarding motion, I just want to tell you, because I find this quite interesting, is that we think of motion mainly about one type of motion, which is you have a filament, microtubule or actin, you have a motor protein that is recognizing a cargo, an exosome, and is moving it along their specific filaments, so myosins for actin, kinesins and dynein for microtubules, and they move that. It's like a car in the city, they know where they have to go. But there are many other motions in cells. One of them that we've worked a lot on, one of them is advection or flows, which is basically this motion of motors, especially kinesins, are dragging the viscous cytoplasm with them, a little bit like a car dragging the air with it. And that motion has directionality, so it is a persistent motion, and it is helping things to be localized as well. The other motion that happens a lot in cells that is not so much understood is active diffusion. That is not persistent, that is random, but it is still ATP dependent. And it's a bit, active diffusion, you could think of it, and it's often a result of cytoskeleton moving and motors moving in a more random manner, but it's been shown to be important for counterbalancing the effect of a crowdiness of the cytoplasm. Cytoplasms are full of stuff, and they get very crowded, and you may have heard about phase separation. Anyone knows about phase separation, this new field? No? Yes, because it's important for neurodegeneration as well, it seems. So basically, our cells need to have a good balance of what it is in a liquid state or in a semi-liquid or in a more solid state, and things that are unbalanced so that suddenly get more solid, like aggregates, that is wrong for cells. So this type of aggregates may come up as a result of a lot of crowdiness. Motion helps these aggregates not to form. So again, motion helps your cells to be healthier. Again, like with us, if we move, we are healthier. If cells have things moving inside their body, they are also healthier. And we did a lot of work with especially physicists, so it's very interdisciplinary. It's a biophysics field, to find out that in the Drosophila oocyte, which is in oocytes in general, large cells, round, you have all these motions happening. You have the cargo-like, and it's very important for embryogenesis that you form an oocyte with the cargos being, I keep on moving it. Cargos going to the right place in the oocyte. They're not moving now. Let me see if I can get the movie going. Yeah. And you have kinesin 1 is a major microtubule motor that is required with the microtubules to move several cargos. And I'm not going to go, there's a lot of work, not just from our lab, but many labs. This induces these flows, this dragging of this viscous cytoplasm with the kinesin. And these flows are important for the advective motion or for advection of a lot of components in that cell. And that we show as well. And then we show that active diffusion, the more random one, just moving around to keep things healthy, is of vesicles, is because of cytoplasmic filamentous acting and myosins there are creating these forces. So it's very cell biophysics characterization of what happens in the Drosophila oocyte. And some work from other labs in the mouse oocyte have shown that this is also true. Now, regarding more diseases, I told you that these motions are not only important for the function of a normal cell, but it has also been linked to diseases. And one very specific case is Alzheimer's disease, which we just heard from Yanis. Thank you. And what has been shown is that both tau, which is a microtubule binding protein, and the amyloid beta aggregates, they both impact on motion in neurons. They impact on how things are moving up and down. They affect the transport of things in neurons. And it's also been shown that microtubule motors, and you have an example there of a just paper published in Life 2024, but there are other examples. The levels of motor proteins are affected in dementia patients. So it could be that you have, as a contribution to neurodegeneration, less transport, less motor proteins, less motion may contribute to that. So we wanted to study this, not only because of the disease, and I'm trying to give that angle because of the audience here, but also because it's not much known about motion in neurons. Okay, now I don't get it. It's not moving. Shall I escape? Perfect. So this movie hasn't worked when we transfer it, so let's try now. But it's basically mitochondria it's mitochondria going up and down in a live larvae. So in the flight, you can look at axonal transport in a live larvae. And you will see things going up and down really nicely along the axon of the neuron. And what we want to understand, and we don't have data on this, is whether these findings of the complex motion and cytoskeletons that are driving the motion in the oocyte, whether it's true in neurons or not. Very different cell type, from a round, large cell to a large but very elongated cell. And the axons, imagine they can have a meter on us, but think of a giraffe or an elephant. I don't know how long axon. I don't know how long the longest axon is, if anyone knows, but it's probably several meters. So how do you move things up and down, how other type of motions contribute to the function of neuron is not really understood. What we have been able, and I told you I don't have a lot of data on this yet, this is a difficult question to study in neurons, but we are interested in that. What we have been able to show is that if you use flies with this humanized trick that I told you about, so you get flies expressing a pathological amyloid beta from humans, and you express them you can drive the expression of this human amyloid beta, whatever you want. We chose motor neurons because of the neurodegeneration link. But again, the genetic power of the fly allows you to manipulate things very tissue, cell, and time specific manner. And here you will see again a movie, but it's not there. At the top, you have the chemographs of those movies, and basically, which is just showing things moving. The top is showing you a control, and you see a lot of things going up and down, while in the amyloid beta expressing flies, human amyloid beta pathological peptide, you do have a huge defect in transport. There is an increase in stationary mitochondria and a decrease in motion mitochondria. So that we can recapitulate. Having a pathological amyloid beta, I wouldn't call this an Alzheimer's disease model because flies don't have, for example, cortex, but you could call it an amyloid beta toxicity model and trying to understand the link between toxic, and this could be also done with a tau form if you wanted, a pathological tau form. How is that affecting motion in cells? And in this case, you could think of the exosomes, what is happening to them as well, right? Are they maybe not being moved to the cell body and then they are more being exocytosed or not? I don't know if anyone is looking at that, but it's an interesting.
It doesn't depend on action. The exosomes do not depend on axonal processes.
Okay.
Local, so we move to the cellular body to be thrown out.
But how does everything go to the multivesicular body? Yeah, but something has to get it all the way to the, not the exosomes themselves.
Not the exosomes in the axon.
Okay, okay.
But you're absolutely right.
For the rest.
Cytoskeletal,
They have to get there. Yeah, exactly. So the other thing that we could recapitulate is that what happens to the neuron if we eliminate the motor proteins? Remember, I told you that maybe reducing motor protein affects of neurodegeneration. So we wanted to see what happens to the neurons in the larvae, but really in an alive animal. So what we did was mutate the kinesin 1 or the kinesin 3, which are the two major microtubule motor proteins in neurons. And we could follow the neurons. That's the nerve cord in the fly, and then you have axon, neuromuscular junction, so synapses with the muscle or dendrite. You can detect which neurons are mutant because they express green fluorescent protein, and only the mutant neurons express green fluorescent protein. And you can then check in my larvae, which grows, it undergoes a series of phases, L1, L2, L3, doesn't matter. If you look at early larvae, we have these mutant neurons that we just induce genetically. If we look at larvae 3, these neurons are gone, and that's what it's showing. In the green one here, I can do that. You can see that L2 versus L3 versus L2 ratio of neurons in control, they're there. Whatever L2 larvae 2 had, larvae 3 still had. If you don't have kinesin 1 or you don't have kinesin 3, there is a drop in the number of neurons with this GFP. Even though in the same animal, they were there before. So they are disappearing, they are degenerating. And now the question then, what we came to our head is, if amyloid beta toxicity blocks transport or motion, and neurons without motor proteins die, could it be that overexpressing motor proteins in an amyloid beta toxic neuron rescues that phenotype of the amyloid beta toxicity? And the answer is yes. And the way we did this is a very simple way to do in the fly, is with the climbing assay. So basically, and this is very well-established technique as a readout of neurodegeneration, which is motility, locomotion capacity. The flies that express the that express the human toxic amyloid beta peptide are on the right. The control ones are on the left. And you can see that they are unable to climb up or less able to climb up. And you do a performance index over many flies and many experiments. And you can see on the right that the red, which is the amyloid beta flies, they drop their performance index with time. So it's age dependent, which is amazing, I thought. But while the ones that are expressing this peptide plus kinesin 1, so overexpressing kinesin 1, inducing more transport, or kinesin 3 in purple in the next one, they can rescue, this overexpression can rescue the climbing capacity of these amyloid beta flags. Okay, suggesting that overexpressing motor proteins may help neurodegeneration. And if you remember, I also told you that it's been shown that the levels of motor proteins in dementia patients are going down. So it could be thought as a therapy, but I'm not gonna do it, but someone may think that increasing transport may help then patient at some point. Now, is this true in human cells? And that's the question that we do want to answer. And for that, we join efforts with Rick Livesey, and you can see, I forgot to say, this is unpublished work, it's in bioRxiv. With Rick Livesey, you can see there just next to my name. And what we saw is, let's use human iPS-derived neurons, and you can see there the protocol on obtaining them. We are looking at neurons on the day 19 time-wise. And what this system allows you to do is basically get this cortical neural rosette that you can see there. And it's a nice way to look at how healthy these cortical rosettes look like, how much interactions or network from rosette to rosette you have in an axonal-dependent manner, many measurements. We did look at which kinesins are expressed, which is on the right. This is, I'm gonna go, don't worry about the details, because humans have more than one kinesin 1 and more than one kinesin 3. So we had to find out which one we wanted to manipulate. And we found that KIF1A and KIF5A are the ones that are expressed and that we decided to look into. And as at the moment, I just want to say that. So the first question we wanted to answer is, are these motors required for neuronal survival, like we saw in the flag? That's the first question. And the second question is, can we overexpress these motors in a Alzheimer's disease model using the human iPS-derived neurons and see whether we rescue the phenotype? And I'll tell you the results of that. The first thing I want to say is that, of course, in this workshop, we are again looking at another model, which is primary cells or organoids. And again, these are, remember, they are reduction in small systems. They are not a human-like completely, but they're extremely powerful for a lot of the human events that the humans, that we display. So it's still very powerful. I think there was a speaker at some point that said, there is no perfect model, but all models are, something like that, that you do get a lot from different models. And the fly as well is very powerful. Animal models are powerful, but organoids are really impressive, I have to say. Oh, a bit dark. Is this the maximum color? Yes, it is, yeah. So you cannot see the images very well, but you can see the quantification. So we did reduce the levels of KIF1A and KIF5A in these human iPS-derived cortical rosettes. And we could see that, yes, there is less number of cells, that's the one on the left. And yes, there is less rosette formation, so what they call clamps. There is a reduced number of clamps, a reduced size of the clamps, and an increased number of free space between clamps, so less connection. So definitely, as in the fly, and it's not surprising really, reducing KIF1A and KIF5A affects neurogenesis. We don't know yet, and we are looking into this now, whether it's affecting neurogenesis, so neuronal progenitor becoming neurons, or neuronal survival once you have a mature neuron. And we are looking into this right now. We then try to see whether we could rescue the pathological situation, Alzheimer's disease model using this human iPS-derived neuron. The answer is no, unfortunately, yet. Always remember the yet. I told someone before, always say yet. We look as a system, trisomy 21, so 3 copies of the chromosome 21 model that Rick Livesey had. And this is a complex model because it's the whole chromosome 21. It's not just APP being 3 copies, which actually it is because APP, the amyloid beta precursor gene, is in chromosome 21. But there are many other things wrong with it. But we did look at phosphorylation, hyperphosphorylation of tau, and levels of peptide, a ratio that is used as a readout of neurodegeneration. And we could not see rescue in this model system by overexpressing the motor proteins. We have now decided that we should look at a simpler assay, not trisomy 21. And Rick has published a paper where he looks at early onset Alzheimer's disease phenotype mutations in the APP, which is the original amyloid beta precursor protein, and in presenilin, which is one of the enzymes that cleaves APP to make peptides. So we're going to explore that. I don't have data on this. And then again, can axonal transport help these diseased models and could then be considered as a therapy for patients. This is the part of neurodegeneration, and we'll carry on working both with the fly and with the human iPS-derived neurons. Now, the second part is what I told you about epithelia organization. There are different epithelia in our bodies, and it's important that epithelia are that way for their function. And one epithelia that we've been looking at in the ovaries is the follicular epithelia. It's a somatic cells that surround the oocyte, like our somatic cells, and it's essential that you have proper organization. There's a lot of signaling with the germline for an oocyte to become an oocyte, for an egg to be laid and fertilized, and an embryo become an embryo. And we've been looking here at an unusual cytoskeleton that is not much known about, which is the spectrin cytoskeleton. Macrotubule acting is quite a lot known about them. The spectrin cytoskeleton is not so much known about it. It's an essential skeleton in red blood cells, but it's not known so much what it does in other cells. And we look at the alpha and beta spectrin. You can see they are formed apically and laterally. This cytoskeleton is formed attached to the membrane apically and laterally. And we look at what the function of this cytoskeleton may be on the columnar shape. You can see how columnar these cells are of these specific epithelia. And we also look at integrin basally because they've also been suggested that they may function in cell shape. And this is all published. I'm not going to tell you much about, but we did see that mutating both spectrin, and you do that again, there are genetic tricks to obtain just a bunch of mutant cells lacking GFP there, and you can see they stay columnar compared to their neighboring cells, which are with GFP, and so they stay cuboidal compared to the columnar neighboring cells. And also looking at integrin mutants, and we apply, genetics, fixed sample, movies, lots of different analysis, and we basically show that integrin in the basal membrane and alpha spectrin in the lateral membrane are regulating actomyosin contraction for these cells to columnarize, to extend and become columnar. Now we wanted to see whether this is also the case in a human epithelia. And one columnar human epithelia is the neuroepithelia, okay? The neuroepithelia, so very early stages of development. This is the epithelia in the neural tube that it forms in very first few weeks of the embryo. And I have a slide showing a bit more about that. It's been shown that the columnar cell shape of these neuroepithelia is important not just for development of brain in general, but important for development specifically of the human brain. This is a Cell paper, an amazing work by Madeleine Lancaster in Cambridge, who we talked to a lot a couple of years ago. And they basically compare organoids, iPS-derived neuroepithelia from human iPS or from other primates, iPS cells, and show that cell shape changes that are unique at the human may be linked to what make the human brain unique compared to primates. So really work, beautiful work. It's a bit hypothetical still, but they did recognize a different stage, that one in the middle, doesn't matter the details, showing that you have an extra step on cell shape columnarization in this epithelia in humans. And just for you to relate what we are looking at in the organoid, again, is reductionist. It's not exactly looking at neural tube, but just for you to see, this here on the left is a cross-section of a developing embryo. I think it's week 2, week 3, not very late. Neuroectoderm at the top, it will be folding, and then it forms the neural tube at the bottom. This is happening all along the animal, from the anterior to the posterior of the animal, of us. The anterior part will then develop this neural tube into the brain, and the more posterior part will develop into our spinal cord. And neurogenesis happens all in this neuroepithelia, okay? for many, 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 many days, but creating, of course, the huge number of neurons that we know. Cerebral organoid labs are now recapitulating these stages. I'm interested in the very, very early neuroepithelia stage before neurogenesis takes place. Many people are studying neurogenesis as well, including Madeleine. So we are looking at these cerebral organoids. This is, again, the same paper from Madeleine Lancaster lab. And we are looking, our interest relies on these very early stages of the neuroepithelia, which is representing a cross-section of the neural tube. You can see the neural tube cross-section at the top. So I'm cutting the animal like that. Anterior will be towards you, posterior at the back. And then at the bottom, I represent these rosettes, which are a columnar neuroepithelia, similar to the one in the neural tube. Again, reductionist. And what we are doing is looking at creating these organoids in the lab, staining them with different things that allow you to analyze cell shape, cell division, apical to basal size, nuclei position, because interkinetic nuclei migration has been shown to be important also in neuroepithelia. We analyze different features of that. And again, just for you to see that there are many aspects of the real neuroepithelia that are recapitulated in this neuroepithelia organoid, including the position, for example, of dividing cells. Dividing cells move apically, you can see there, dividing neuroepithelia cells. And it's the same in our organoid, we find dividing cells only in the lumen area. So together with Natalia and Julian, also in Queen Mary, we've been doing three things. One is trying to identify several patterns of this tissue architecture of this neuroepithelia. And instead of doing manually, because as you heard, there can be lots of differences in control organoids, we are using machine learning a little bit to extract patterns that are reliable. Again, how long the cells are, where are the nuclear position, what is the curvature of this epithelia, and so on. We are also manipulating either genetically by CRISPR for the spectrin skeleton, or in the case of integrins, we are doing blocking antibodies. And then we ask the question, what happened to that? And we have very preliminary data that I'll show you in a minute. The other thing we are doing, because of our interest with biophysics, is trying to measure forces. Where are the forces of these neuroepithelia coming from? Where is the tension coming from? And are these important for development? And we do that by using oil droplets, artificial cells, that get incorporated into the neuroepithelia. And they are very good measurement of how things get pulled in different places, like oil in the middle of I don't know how to say, maybe in a little of water. And then if you move the water around, this oil kind of moves around and you can use it to measure what's happening to the water. And we have some data. The first data is eliminating a spectrin in this columnar neuroepithelia is affecting nuclei position, which is known to be important for the architecture. This is all we have at the moment. We don't see yet defects in columnarization. So this may be a function of a spectrin cytoskeleton that is not conserved. So it's important to know that. But it seems that a spectrin is still important for the tissue architecture of a columnar epithelia. And the other data, we leave that one there, is that for integrins, the first experiment we've done, you can see in control rosettes at the top. There are 5 or so clear rosettes there. And if we incubate these organoids with an antibody that blocks integrin function, we don't see rosettes, which is not what we want. It clearly says integrin are important for rosette formation, neuroepithelia formation, neural tube formation. But we would also like to allow some development of the neuroepithelia. So we will incubate later this organoid with the antibody, blocking antibody, and ask the question, what happened to the architecture of this neuroepithelia? And regarding the forces, we've also seen that there are compressive forces resulting from a rapid cell proliferation in these organoids, and that is extruding this artificial cell outwards. So there is a force outwards. And that's it for the science part. Do I have a minute to, yeah? Right. I just want to say one more thing, because this is how our collaboration started. And as you know, I'm the fly lady, that I set up together with Marta and Lola over there, a charity called Rosafrica, which is basically helping research in Africa in any way we can, using Drosophila as a model system. And we've been doing this for many years, extremely successful and wonderful, wonderful charity. And we do lots of different events, and now we collaborate with other organizations that have been created during the years that we've been helping them. in Nigeria, in other countries. And the idea is to help that map to change a bit. You know, if you look at the African continent in population compared to scientific publication, the surface area drops dramatically in the scientific publication compared to, of course, population. And what I wanted to say, why I wanted to talk about it, apart from, you know, join us in any way you want and help us with anything you want, is that we are joining Merck and other companies and other scientists, Rocco, Vide, across, there are like 4 or 5 hubs, Egypt, South Africa, with some now we will probably talk, Rwanda, Nigeria, and so on, which is to expand this collaboration, not just with Drosophila as a model system, but with any model system that allows you to do functional genomics. You know, you have a lot of variants of African population now. What are they really doing? How are they linked to that disease? And we want to study, or you want to understand it at the more cell and tissue level. Some countries like South Africa and Tunisia could do organoids. Others like Rwanda is really not there yet. There, we probably help with flies. In another country, you might help with zebrafish. So the idea is apply to a big grant with tailoring model systems in 4 or 5 hubs in Africa to try to understand these variants. And I think this slide is telling you that the charity wouldn't be, you know, working without lots of support, lots of people helping in completely voluntarily, both in, I forgot your names, are not there, that's terrible. I put the dots on those, but I should, and both all over the world, but also in Africa. And now a lot of the people that we've been training and helping to set up labs are now joining our workshops and helping others in Africa. And as an example of that, the group in Nigeria is organizing this webinar. If you want to join, you're welcome to join. There are online talks from several scientists, women scientists, trying to discuss what have been or how we have navigated as women the research landscape, you know, our personal life story. It's the first time I'm giving a talk like this one. I have no idea what I'm going to say. But, and yeah, thank you very much to all of you.
Isabel, thank you very much. It was awesome. And you will note later at the end of the session how multidisciplinary you need to be, and how multidisciplinary we need to work together, all together.
Yeah.
I will give the chance to the audience. You have to ask question. And you have to answer within it. Yes. Yes. Bye.
Can I ask now?
Of course.
Where are you? Yeah.
Thank you for the presentation first.
You're welcome.
So
My pleasure.
through this study and your collected data, did you try to create a prediction model from this data, for example, to compare the outcome or the differences between the Drosophila model and the human physiological pathology?
Okay, so you mean trying to use a computer-based model, is that what you're saying, to understand the finding for neurodegeneration, you mean?
Yes, and the accuracy too. I wanted to compare the accuracy between the Drosophila model and the actual human pathological physiology.
With data that are known, I mean, I guess you have to choose one model. Let's say the amyloid beta toxic model. Is that what you have in mind? Okay. And then, yeah, we haven't done it. And I don't know whether anyone has compared at the pathological level or the physiological level, what is different between the flies, this neurodegeneration, and the human. What I can tell you is the link between, which is a bit what Janis was again talking, the link between the cell biology, what goes wrong in the cell, and why that neuron dies is not really understood yet. What is first, right? How is hyperphosphorylated? Why is it hyperphosphorylated? What does it do then? And how is that doing whatever to the microtubule, because it's a microtubule binding protein, results in axonal death or in neuronal death? That is not really understood at all. Not in humans, not in flies. So in flies, one problem that we have, and it's in general a problem, I think, in all model systems, studying aging is really difficult. and link into cell body. So you can study aging in the fly like I did for the climbing assay. Really easy. Your flies, they live around 50 days, and around 20 days, they stop climbing. I cannot look at the cell biology of these flies. There is no system yet that allows me to look inside the neurons in this adult fly at the level of what I need to know to understand what amyloid beta is really doing in that brain, in that neuron. It's not known. Human cells, there's no aging system. There's no aging system for organoids. There is no aging system in mice, really. That you really, there are models that artificially induce an aged system, right? That can be, but it's not, there are very few where they look at a natural aging and then see what happens there. So that's a caveat, a gap in, I think, any model system. But yeah, informatics could help, and I don't know much about computing, but if anyone has a comment on that, yeah.
Do you think the prediction model will be helpful to solve that issue?
Prediction of what?
Exactly. I think that's the problem. You have to be precise with what you want to predict. And also one careful thing about prediction models is it's very good to do modeling. And I have tried it for example, the epithelia work. That we've tried to put together the spectrum forces, the integrin forces. And you do really have to you can come with any model. You can have 10 different models. But what is really reflecting what is happened? So what you have to do in that case is create a model, predict what would happen if you now mutate X or you overexpress Y, and then see whether the result in your tissue and the model comes up with the same thing. Only then I would trust the model. But in your case, I mean, you may be onto something, but you have to be more precise on what you want to predict. What is what you're trying to understand?
What you feed the model in order to create a model, you need to feed some data. So
Yeah.
if you use your own proteomic data or whatever, then you say, based on that, predict me that where this is binds or where this comes.
Yeah.
You need to feed it. So since
Yeah.
that's what you need to
So
say.
it also depends on the type of data.
Absolutely.
Exactly.
It depends on the type of data, on the question that you want. But the best way is sit with computing guys, talk about the process, try to find out. And neuronal networks are very attractive as a
Yes.
computing network, right? And there are many people doing it. I'm not into it. And again, remember, I'm like 2 years into neurons and organoids. I'm really very young, a baby. So, but yeah, I'm sure there are people doing this. Yeah.
Thank you so much for this amazing presentation. I have a question about blockers or therapeutic agents. Have any blockers have been developed to effectively target the aggregation or toxicity of amyloid beta and of tau proteins into the prevention of neurodegeneration diseases?
I think Yanis probably can reply that much better than we did. So he did mention that there have been efforts. He had a slide with amyloid beta aggregates, tau, and but he can tell. Yeah,
To prevent what?
the aggregation.
Aggregation of amyloid beta or tau. Okay. Yes, and they fail. So they fail to stop pathology. For example, you know now
Vai, yeah, Yanis.
it's rude to. There are monoclonal antibodies that they are as a drug in the States. Monoclonal antibodies that they bind to amyloid beta and reduce it. They really reduce, so they help the microglia and macrophages remove the amyloid beta from the brain. That works very well. However, there is no improvement of memory in this patient. So
And also works very well with the earliest
yeah,
stages.
and also
Very, very early,
But works in the clinical output. So the answer is no for amyloid beta. For tau, they are using some anti-aggregation blockers, but so far there's no clinical output. What they are using now is antisense oligonucleotides to bind to mRNA of tau and reduce total levels of tau, that's avoiding toxicity through tau. Again, there was a Nature paper 2 years ago, 1.5 years ago, showing that these answers from Biogen reduce the tau level, tau PET imaging in the brain of humans, AD patients, but there's no the clinical readout yet. Or it failed, or it needs some more time.
Yeah.
Okay, I have
there.
In the back, and I have
She was.
myself, yes, in the back. I have myself question for both of you. Before, sorry, my friend, I will start myself.
No, give a chance to the audience. You talk to the.
I talk too much. Isabel, allez-y.
Yeah,
Thank you for the presentation. Artificial cell,
yeah.
it's related to an artificial intelligence.
No. This is my collaborator, Julian. He's a bioengineering. Basically, when you want to understand the tension of forces, so it's been now shown that physics of a tissue is very important during development and during disease.
It's a model.
It's an oil
Oil.
droplet, oil, so lipid layer
Mm.
that is functionalized. So they cross-link a few layers of things so that you end with cadherin, which are adhesive molecules that then bind the epithelia cadherin and they get incorporated into the epithelium. And then because they are passive, they're just oil, there is no contraction inside, there's no actomycin, there is no microtubules, there's no motion. They respond to the forces of the other epithelia pulling them, and the deformation of the droplet tells you where forces are happening in that tissue and whether they are affected in a disease. So people are using it for that.
Or no, organoid. The motor neuron.
So the cerebral organoids are being used now or iPS-derived cells are being used to look at neurogenesis, different type of neurons, including motor neurons. Yes, absolutely. Yeah, then you can.
Yeah.
Good morning.
Hi.
Thank you for your presentation. I have two questions.
Yeah.
The first one, what is different between 2D and 3D organoids?
Yeah.
And second one, what are human organoids used for?
What an organoid is good for.
Yeah,
Okay.
used for.
So, I'm not an organoid
No.
person, but so what do you think a
With
2D
that,
and a
yeah.
3D organoid is?
Yeah,
What do you think?
yeah.
Yeah, exactly. 2D system means just X, Y. So you have kind of a flat group of cells. So, and then 3D means that you expand on the cell. So the one we look at, both the neuronal clamps and the neuroepithelial organoid, they have a set axis that the others don't. So it means it's increasing the complexity of that tissue. Our tissues, the epithelia are kind of 2D, but then they are interacting with other cells in any case, and then they also fold, and so there is vertically nothing that is just flat. It's a bit like when people say the image of a cell in a book, almost no cell is like the one in the book, right? So almost no cell is, or no tissue is like a 2D flat with nothing. So to mimic a little bit more our sense, they went to 3D. Yeah.
Okay.
But I'm new on this.
So last question, and then we will go to the coffee break. Just 25 minutes because we will start with a talk, fantastic, from Cambridge University and Katja.
Yeah, she's
So the
fantastic. Yeah.
Last question for both of you. So the story of diabetes and Alzheimer's disease, Alzheimer type 3, called now, I think. No. So how it affects the traffic of lysosome and how it's also, affecting
You
the
mean the traffic of exosomes?
exosome. Sorry.
By the way, lysosome
And the-
trafficking is very
And the
linked
traffic
to
of the tubuli. So what?
diabetes. Or-
Yes. So the diabetes, the fact that you have more glucose, how it affects the traffic here, and help it by the
Okay.
kidneys in here.
So what is the link between diabetes and Alzheimer, Mary, at the cell biological level? Is it known?
Diabetes and signaling, and insulin signaling is known to, help both amyloid beta and tau, but help at the cellular level. So the signaling, the insulin signaling inside the neurons and microglia, they help the amyloid beta reproduction and tau population. But and then I see, for example, work with insulin molecules, insulin signaling molecules inside the exosomes. But I don't think they affect, because exosomes is like vesicles that they are flying around. Until they release, they need some cytoskeletal support. But after, they go around.
Yeah.
Through the blood, go around. They don't need any cytoskeletal
Outside of
stuff.
outside of
And
the
they
cell.
also don't, they're not new organelles. So their cargo, their transportation inside the cell has nothing to do with mitochondria or other kind of cargo. It's just from the multicellular body, they are going out, that's all.
Okay,
Stop there.
But they do.
axonal, no axonal involvement, no neuronal axonal involvement. Don't confuse these two things. Now, outside the neurons, they're floating because they're neurons. They are in the blood.
Yeah, so regarding the diabetes, I don't know what the impact is on
okay.
the cell biology.
So-
But one thing I want to add is tau is a microtubule binding protein, and amyloid beta is, the peptides are the ones that aggregate. These peptides come from a protein, and maybe you know more, the APP protein. What is this protein doing inside the cell before the cleavage? How is that related to Alzheimer's in any way? And